Okay, welcome everybody. So I think we are ready to start. So here we have Mario Garrido. He is associate professor from the University Rey Juan Carlos. He has collaborated a lot with mosquitoes people here, with Josue, with Mari, with this research group. He's going to talk about the dilution effect of the biodiversity on the disease risks. So welcome, Mario. And the floor is yours.
Pleasure. Thanks for the invitation. I'm delighted to be here. I just realized that it must be in English, so I'll try to defend myself, okay? The best I can. So what I want to try to explain today in detail is my own experience with the famous relation between biodiversity and disease risk, no? And what is crystallized in the famous dilution effect that was proposed in the year 2000. And I'm going to introduce first a little of the theory about this relation between biodiversity and disease risks. Then we will land into the dilution effect, and I will combine it with some of the results of my experiments or field studies, whatever, in order to get to a kind of understanding of what is going on there, what is the debate about. You know that this is one of the more controversial issues today in this ecology field. And I think it's interesting to make a little review of this relation of disease risks and biodiversity, okay? So thanks very much and welcome. As you well know, all of us, in the last 20 years, mainly since 2003, we are experiencing a dramatic increase in the number of epidemic outbreaks of emerging infectious diseases. Just starting from the 2003 with the famous SARS outbreak. Then we go to the swine flu around 2009, the Zika epidemic. Also, we experienced not time ago the Ebola outbreak and the famous Excalibur dog, if you remember well. We finished just time ago, not finished, we are still suffering some kind of epidemics, but the big one, you remember the COVID-19, with almost 7 million people dead already and still new cases that are to be confirmed. What I wanted to show you, I'm sorry, is that not only we have a great increase in the epidemic outbreaks in the last 20 years in the number of events, but also in the total infected cases and the number of deaths that we record all over this year. You know? What we know about this epidemics break? What we know about these emerging infectious diseases? So from one part, we know that in the last 60 years, as we can see in this graph, not only the number, the absolute number of these AIDS are being zoonotic, but also the proportion of the zoonotic AIDS are increasing. As you see in the last decade of the 20th century, the proportion in white of the zoonotic diseases coming from the wildlife are really increasing a lot. We can say today, or what we know today, or what we can estimate today, is that 75% of these new emerging infectious diseases are mainly zoonotic. And changing the view from the other viewpoint, we can say, or we are estimating that 60% of the new infectious diseases in humans are zoonotic in this case. But not only zoonotic, you also know that are vector-borne transmitted. If you see the graph, the figure, the tendency is more or less similar to the number of zoonotic AIDS. The proportion compared to the rest of the kind of transmission is mainly vector-borne, as we can see also in this graph, where we have here the cause of the new emerging infectious diseases, and we have here the transmission, how these diseases are transmitted, and we can see here that most of them are transmitted by vectors. So we have these AIDS that we know that are mostly zoonotic and are transmitted by vectors, but also we guess, or we are more or less sure, that more of these new outbreaks, new epidemics, new emerging infectious diseases are related with the human activity, with the economical and social change that are disrupting some kind of the equilibria in the ecosystems, and in this way, we have to afford to be in contact with new pathogens. Okay, some of the most related consequences of the human activity are more or less schematized here. You know, forest fragmentation can put us in contact with new diseases. Then we have this kind of intensive livestock farming or the treat of animals that are making the pathogens to make new recombinations, new kind of recombinations. And one of the main consequences is biodiversity loss. That is the point of our speech today. For me, for my taste, there is still one thing that is not here that should be represented, that is the great amount of flights or movements or translocation of people from one country to the other in the 21st century that are contributing to all the time to be sharing all around the globe this kind of new epidemics that are moving from one part to the other. But mostly here are represented, mainly are represented the big consequences of the human impacts and how they can affect these ecological systems. Okay. As we all know, the host parasite systems are ecological systems, and as all ecological systems are really very complex, are integrated of very different components and the relation between them. So the question is, all these kind of activities, all these kind of human impacts, how are affecting my host parasite systems? So from the origin, from the beginning of the studies of the disease ecology systems, okay, we know that we focus mostly on how these kind of activities impact on the host, impact on the parasite, and mainly on the relation between them. This was one of the most, let's say, traditional way to approach the study of host parasite system. But of course, with the years and with the new analytical tools we have, statistical tools, etc., we are now getting to integration of also how they impact the vectors and how they impact the host community composition. And this is the main focus of the talk now, how these kind of activities can affect the host community composition. That is other way to say how it's affecting biodiversity. Okay? Here is where it appears one of the main usual aspects, how the biodiversity loss is affecting the host community composition and how this can contribute to increase the disease risk perception. Okay? This is one of the focus of our approach in this case. But first of all, we have to do review of what's going on. We are really in a moment that we are losing diversity all around the globe. So is biodiversity loss true? If we go to the figures, it seems that that of course is true. If we go to this panel in the left in the top, this is a representation of all the group of species disappeared in the last 5 centuries all over the world. Okay, and here in the bottom right, we have a representation of how is the state of the different populations of different species all around the world of different groups. We have vertebrates, we have birds, we have amphibians, reptiles, etc. As you see over there, there is only small cases when these species are increasing, is this green marks over there. And if we forgot about the unknown situation of these species, what we can see is that the majority of the population are or decreasing or remains stable. Okay, so okay, we can say that this is true. No, we are in a kind of biodiversity loss all around the world. What we can start to call, what we call from time ago, the sixth extinction, the sixth global extinction all around the world. So what we see until now is that for one part, we have a great biodiversity loss, and from the other part, we have an increase in emerging infectious diseases. The question is, how are they related, and if this related is correlational or is causal? Because it's not exactly the same. We can see, of course, that biodiversity loss is increasing, emerging infectious diseases is increasing, but what is there in them? Just as an example, if we go to a global scale, we can see in the panels of the top that the geographical variation in pathogen richness and the hotspots for emerging diseases are the same as the hotspots for erosion in mammals like biodiversity. So in a global scale, we can say in some ways that they are negatively related, okay? This relation that is not, let's say, new, that is something that 100 years ago, even more, some people even proposed, is what Osfeld and Kissing in this paper in the year 2000, working with, they stated, or they put conceptual framework for this negative relation. This is what they call the dilution effect. In essence, what they said is that biodiversity is expected to be linked to reduced or wasted transmission. The take-home message, really, what is that American people like a lot the take-home message? Biodiversity buffers infectious diseases. Of course, this is a very, very nice message to take home. All the people were, how can I say, all the people were like receive this message in a very good way, because it's kind of utilitarian message that if we save the biodiversity, the nature is going to save us. Something related, if we have to put some kind of anthropological meaning. Okay, so until the year 2012, almost all evidence that we have is like according to what Osfeld and Kissing proposed. There is a negative relation between biodiversity and disease risk. Indeed, there was it's not here, but there was a meta-analysis for one in 2016, I think, that states that 80% of the publications up to this moment really defend the dilution effect. They found some kind of dilution effect, okay? What's going on? The press media really embrace it, accept it very well. Why? Because what I was saying, it's like a kind of Panglossian message. Let's say that it's a kind of Disney message, okay? Like, save the nature, the nature is going to save you. So, without any kind of critical argument, in most general press media, we find, even in some kind of organizations like Ecologistas en Acción, we find that they really accept it with delight, not the dilution effect. But from 2012, the debate started to open, and it started to appear a lot of publications from really very good researchers like Emil Lafferty, putting in doubt what the dilution effect states. Indeed, some people like Randolph and Dobson state some serious correction for that. They said that all this literature up to 2012, they were like doing an incorrect data interpretation, they were doing incorrect hypothesis testing, experimental design, or even more, they were telling that there was some kind of bias in publication when we were only publicating results that really confirm the dilution effect. But it is really not like that, okay? So up to today, we have a very big debate, as you know, a very controversial debate. Some people state that there is a dilution effect, there is a negative relation between disease risks and biodiversity, and some people say that we have really an amplification effect. We have a positive relation with biodiversity and disease metrics. Indeed, the sentence that can summarize what the amplification effect people think is that biodiversity begets biodiversity. The more hosts we have, the more chances to have parasites adapted to them. So also it's has a lot of ecological meaning. No? It's really true, the case. What is more prevalent? Really, up to date, we don't know. What we don't know is that not only the two options are the dilution or amplification effect, some people modelize things much more complicated. What is, in my opinion, more realistic to what can be happening in nature, okay? So, at this time, when I started to read this paper, I was in my postdoc in Israel, in the desert Negev, and we have a very good chance to try to explore what's going on in these sand dune systems. Why? The Negev desert with Egypt is a system of dunes composed by rodents communities, gerbils communities, where we can find also this kind of different kind of biodiversity that can contribute to test this kind of dilution effect hypothesis. Okay. Indeed, what we have, we start to work with a system that was composed by rodents, two kind of pathogens, and then a vector. What we can find here is this spot in the Negev desert where we have all these plots distributed in distance one to each other, where are composed by these three different gerbils. There are, of course, like very related species of gerbils between them. So we have some plots where we have only Gerbillus andersoni, the most common one. We have other communities where we have Gerbillus andersoni with Gerbillus pyramidum in all this part, the biggest one. And we have also communities where Gerbillus were present, as you see, mostly in community with three of them. So we have some kind of living laboratory system where we have different replicates. In some replicates, we have just one host, in other we have two hosts, and in other we have three hosts. All of them phylogenetically related between them, so it's very nice to see if they share the pathogens or not. From other part, we have two kind of pathogens in the blood mainly. We are working with erythrocytic cells, and we have two kind of parasites, Mycoplasma and Bartonella. And one vector flea that, of course, we know that is transmitting Bartonella, and there are a lot of chances, we have still analyzing these results, that in some way they can transmit Mycoplasma also. This is another word. Okay? So we decided to go ahead and recover some of the blood samples that we have from gerbils from a lot of time ago to see what's going on over there. So we take samples that were frozen from many years ago, I don't remember how much, and analyze the blood for Bartonella and Mycoplasma. As you can see here, we have only data for Gerbillus andersoni and Gerbillus pyramidum, the green and the purple one. Why? Because we didn't have enough Gerbillus, that is less common, so statistically was not meaning. Our first surprise was to see that while Bartonella was distributed similar between the two species, both Gerbillus andersoni and Gerbillus pyramidum were very much infected, and there were no difference between species. What we found for Mycoplasma, the first case is that in Gerbillus pyramidum we found a very low prevalence, while in Gerbillus andersoni we found a prevalence up to 90% more or less. So we say, okay, let's go again to the field, to the sampling systems, to take new samples, and we can see what's going on. We sample different plots with only Gerbillus andersoni, plots with only Gerbillus andersoni, plot with Gerbillus andersoni and pyramidum, the purple and the green, some with Gerbillus and Gerbillus pyramidum, that is over here, and the other with the three species. And what you see in the bar plots are the prevalence for all over the host we collected in this place. So at the beginning, we said we don't find any kind of relation, and when we start to enter many variables in the model, the variable that explain better the model in this case was the presence of the vector of Bartonella, that is the flea, Sinosternus cleopatra. If you see this black line is the abundance of Sinosternus cleopatra. There is a very nice relation between the bar plots of the infected and the line of Gerbillus. So our conclusion in this case was, okay, for the moment what we have here is a pathogen that seems to be generalist and can infect the three kind of hosts. Then we go to Mycoplasma, and we make our own predictions, no? We said, okay, if Mycoplasma is a generalist, the proportion of infected, if 100% is the total number of infected individuals, I can expect, if it's a generalist, that this distribution is going to be the distribution of the gerbils in these communities. I don't know if I explain myself. As we have more or less 35% of rodents in the community that are Gerbillus andersoni, if Mycoplasma is generalist, I can expect that of the 100% of infected individuals, 35% are Gerbillus andersoni. But what we found indeed was that Gerbillus andersoni was infected. Of the 100% of infection, 80% belongs to Gerbillus andersoni and 20% belongs to Gerbillus pyramidum. We found no infection from Gerbillus. So Then when we go to do the difference between plot, we realize that when there is Yerbillus andersoni only, the proportion of infected go to 80%, what is very similar to here. When we put Yerbillus andersoni and pyramids, more or less. And when we include spots with Yerbillus yerbillus, there were no infected individuals. So we can say, okay, here is working. We have an increase in biodiversity and a decrease in the prevalence of infection. No? We can say that there is a kind of dilution effect. But again, when we put the different variables, the best variable explaining these results were not the species region itself, was the presence for Yerbillus yerbillus. Here is the same plot, and this black line is the proportion of the abundance or the amount of Yerbillus yerbillus in the community. So the more Yerbillus yerbillus, the less infection of mycoplasma that we have. The first conclusion that we have is mycoplasma is specific to Yerbillus andersoni host, and there is a pronounced dilution effect. Okay? So the next questions are, okay, we can accept that there is a kind of dilution effect, even if the factor driving this dilution effect is the presence of one species that is not common in a lot of plots. But what are the next question is like, can a non-primary host exert a dilution effect? Is just the presence of a non-common species what is leading the dilution effect? And from the other, mainly, what are the underlying mechanisms and the causal relationship that makes this chance to happen? Okay? So we go into the literature and try to find any kind of response to that. Okay? What we found in the literature was this paper that is not very commonly cited, but is, I think, one of the keystones in this dilution effect. That is this paper from Kissing and Osfeld, the same that state the dilution effect, and they established 5 mechanisms by which a dilution effect can happen. I'm not going to explain in detail because they are very tough, let's say, but just if you add or eliminate any species from the original population, you can have an encounter reduction between infected and not-infected individuals. Let's say there is a community of infected and not-infected individuals. I introduce one new species and the contact rate decrease. On the other part, we have a transmission reduction. It's not only that they not encounter, but the transmission is not so direct, let's say. If I found a new species and I try to infect this new species, it's not going to be successful. From the other, we have also a susceptible host regulation, some kind of action of this new species over the susceptible hosts that can be infected, that are reduced. Also, a recovery augmentation. The new species can contribute in any case to make the infected individuals to recause mortality induced by the own disease. Okay? So are kind of 5 mechanisms by which dilution effect can happen, or even amplification effect, if we add or eliminate any species from the field. So let's say, we say, okay, so let's see what people have done at this respect. And what we find when we find any kind of experimental approaches to these kind of mechanisms, what we were looking for is like, we were looking for publications that test these 5 mechanisms. What we found, a big research gap about what is the role of the background species. That's to say, in most of cases, these mechanisms were tested into the main host, let's say. But Yerbillus yerbillus in our case was not the main host, was the, let's say, the background species, the species that is in the back. No? I try to represent it here by putting it in gray, what can be this background species. So what we can say is like, okay, in my case, in my system, I have species that are diluters, that reduce the amount of infection, and species that are amplifiers. And we can separate into them. And the background species can be some kind of that. In our case, our amplification seems clear that is Yerbillus andersoni, and then it's probably they will have one diluter that is Yerbillus pyramidum, and the most the more important background species seems to be Yerbillus yerbillus. So we have found this research gap, And we also found other experimental gaps in this research. What we realized, that there was a lot of theoretical work about that, but when you try to to put bridges into these theoretical and experimental studies, we didn't find a lot of we didn't find a lot of them. So we tried to solve them, and then we found also that there is a single hypothesis bias. In most of the cases, they were testing only one of these mechanisms under experimental condition. So what we try to do, a colony in the laboratory with the 3 species that we grow by ourselves, and we decided to try to test 3 out of these 5 mechanisms at the same time, because we're impossible. I will explain then what we cannot test the 5 of them. And we decided to test the transmission reduction, the recovery augmentation, and the infected host mortality. I will explain later how. What we cannot test, because should be to do in the field, is the encounter reduction. We cannot do an experiment about how many encounters they have really in the field in natural conditions, and also the susceptible host regulation. These 2 mechanisms requires the animals to be in freedom to have their normal behavior, and we were not about to do that. It was impossible. We were in a laboratory. So we decided to test the other 3 mechanisms. How we did it? And at the same time, we tested the response of the 3 hosts and the 1 parasite, because in general, almost all the experiments tested or the host or the parasite. So what we did it, we take individuals of the 3 species, 11 individuals of the 3 species, and injected with mycoplasma, and take another 11 and injected with PBS. And then we just follow the infection dynamics every 10 days of mycoplasma in the 3 species, Gerbillus andersoni, Gerbillus pyramidum, and the more gray is Gerbillus gerbillus every 10 days. And at the same time, we follow some of the parameters of the host, okay, of all the hosts, of infected and non-infected, and before infection and after infection, okay, in both cases. The activity, the body temperature, the body mass, etc., etc. So what we found, I'm going to follow one to the result. But first, what we wanted to test it, to see this relation, the dilution effect was working, we tried to go step by step. First, we have to confirm that our hypothesis that we have 1 diluter and 1 amplifier and 2 diluter was true. We take 3 kind of different variables. From one part, we analyze the chances for each of the species to be infected in all the periods of the infection. We have a peak of infection, we have a decrease in infection, and we have a plateau of infection. So what we found here is that for Gerbillus andersoni, almost all individuals were infected for the 3 different periods of infection, while Gerbillus and Gerbillus pyramidum were not. Also, we measured the infection length, how much the infection, once after the first detection, how much time we still detect mycoplasma in the blood of the different hosts. And again, we found that Gerbillus andersoni infection lasts for more time. And lastly, also, in order to see if the amplifier is an amplifier transmitting the disease, we measure the activity of the infected and not infected individuals, the activity of the frequency of movements in the 3 species. What we have found here, that the activity of Gerbillus andersoni, of the infected Gerbillus andersoni compared to the non-infected ones, increase related to the non-infected. That is to say, once infected with mycoplasma, Gerbillus andersoni moves more. So the chances to encounter new individuals are greater, and the chances to spread the disease are also higher. Okay? So our hypothesis in this case seems to be true. We have 1 amplifier and 2 diluters. Next step was to test the transmission reduction. Is there any chance, there are more chances in one species to the other that can reduce the transmission? Here we don't find any kind of difference. The 2 things we measure, that was the probability to be infected after we inject the animals, because at the beginning we expected that not all the animals we injected were going to be infected. But we found that indeed the 3 species were infected. And also the intensity of infection in all the period of infection was more or less the same. So in this case, we can say that we find no evidence that there is a transmission reduction when you include GG or GP in the community. Okay? So for the moment, we have only our general support. The second hypothesis we test is the recovery augmentation. Are diluters clearing faster the infection? And once they are clear, they can reproduce the infection again after a second inoculation. So in this case, we found that when Gerbillus andersoni is infected, it never cleans the infection. But some Gerbillus clean the infection, and all GPs clear the infection. But there is also another thing that for me was even more interesting, that is, after a second reinfection, after a second inoculation of Mycoplasma, there is again an infection or not. And what we found is, like, once Gerbillus clear the infection, they never become infected again, never. While Gerbillus pyramidum, in a third part, becomes infected again. Of course, we cannot do it with Gerbillus andersoni because all of them make the infection chronic. So well, it's impossible to do it. So in this case, it seems that yes, Gerbillus recover faster. So once we're in the field, once it's infected and clear the infection, the next time they encounter this pathogen is not going to be infected, and the transmission is going to be stopped. Okay, so this is one kind of mechanism that is working. And the last one is if there is any kind of increased host mortality in any of the three species. And also for here, we find no support at all. Okay, we find that the change in body mass was the same in the three species before and after infection, and for control and for infected individuals. And also we didn't find an individual death. So also there is no kind, this seems to be that the GG or GP dies more in laboratory conditions when we infected with Mycoplasma. Can be that there is kind of sublethal processes in the nature that we cannot test here because we're in the laboratory, but we cannot forget about that, okay? So this is something. So in general, what we see is like, oh, okay, it works. We have one diluter, we have one amplifier, two diluters, and we have one mechanism that is working. So it seems that the dilution effect in this case is working, and it's logical with the data we found in the field. So in our case, we can conclude that despite all three Gerbiles are hosts for Mycoplasma, the three of them become infected, we have that Gerbillus andersoni is an amplifier, and Gerbillus and pyramidum are diluters. So the question is, it seems that there is a continuum in the Gerbillus andersoni, Gerbillus, and Gerbillus pyramidum. And what we learn from this is that it's not only the biodiversity composition or the biodiversity richness what is leading the dilution effect. So it's more the own characteristics of the host and the parasite, and how they are related one to each other in different conditions. What I want to say with that probably what we can do is to first define what is the role of each species in a community for a particular pathogen to state that there is a dilution effect or not, not only to measure the biodiversity and then say if there is a kind of correlation. Okay, so we have some kind of evidence here, but this is still not enough. We don't have a community approach. We cannot test the interaction between individuals, so it's more or less complicated. Then the next step, and I'm going to be fast with this second publication because I think it's likely more complicated. We said, okay, it seems there is a dilution effect here with Mycoplasma. It seems that it's not with Bartonella. Let's see how works here the last component of this speak, that is how human disturbance can alter these systems. So we went to the same plots again, and we analyzed with photos of the last 50 years, or orthophotographies, I don't know how to say in English, orthophotos of 50 years ago. We can analyze what is the community composition, with orthophotos we can see the last 50 years, the dunes are stabilized or semi-stabilized. As the dunes are stabilized or semi-stabilized, is related with the human activity. I tell you why. In general, we have here the frontier with Egypt. In all this part, there is seeps and grazers all over the place. When the grazers go to the dune, they break the crust, the biocrust of the dunes, and the dunes becomes to stabilize to semi-stabilize. This changed the host community composition. This is our guessing, and this change in community composition can be checked for the last 60 years. No, we have data from this community composition from other researchers that were working years ago, and we have this orthophotos. So we went to use this combined statistical approach, this path analysis approach, combined with model selection approach, and we introduced, I don't know if you know how it works, more or less. You have to introduce here only causal relationship that you know how it works. You make different models with different arrows, and what the model tells you is what is the more the causal pathways, only causal, no correlational, the causal pathways that are more likely to explain my relation between my independent variable and the dependent variables. If it's more common that it goes through these actions or it goes through these actions, okay? So what we find in the result, just to sum up, because it's really complicated to explain, that from one part we have that in the semi-stabilized sand, okay, our main question was, are the global processes, climatic and anthropogenic processes, related to local processes and related to the pathogenic bacteria, okay? So what we found with this kind of models is that in a stabilized sand, the climatic and anthropogenic conditions can affect the heterogeneity in sand stabilization, can make a dune to be semi-stabilized or be stabilized. This change, and about changing the community structure, they can influence on the pathogen occurrence. What we found in this case? Something very logical and related to what we found before. If we go to Bartonella, what we found is that in the semi-stabilized sand, where it's more common that the vector can put X and there is more density of vectors because they can dig better into the semi-stabilized sand, the proportion of hemotropic Bartonella, the generalist, is more common. While in the stabilized sand, Yervilus-Andersoni proportion is greater than in semi-stabilized sand, and mycoplasma can increase in this case, okay? Just to summarize. So there is any effect of the anthropogenic disturbance. In our case, we found that yes, for mycoplasma, there is, okay? But in the opposite way, because a stabilized sand is the more natural one. Semi-stabilized sand is the less natural one. What we found in this case, the anthropogenic disturbance, in this case, the non-anthropogenic disturbance can increase mycoplasma. So it's related, but it's something specific. Here is just a small summary of what we have. We have in mobile dunes, the prevalence was low. In semi-stabilized, increased Bartonella and mycoplasma is variable, depends, because we do a lot of different issues. And in the stabilized sand dunes happens the opposite. Just to finish, I write here some kind of integrated results, and in the last publication that we have a sector right now, that we are going to present in the next Congress of the Sociedad Española de Ecología Evolutiva y Etología, we found also that there is a kind of different interplay. What we tested here, it was the interplay between the 3 hosts and the 2 host species, how they interact one to each other to try to understand what we found in nature, okay? I cannot introduce a lot because I don't have a lot of time, but just to say that, of course, what we have learned, what I learned in my process is that we cannot say that dilution effect is working all the time. What we can say is that we found dilution effect, but of course, what is taking the role of this kind of relation between disease risks and biodiversity is the characteristics of each specific host in the community and how they are related with a specific pathogen. But we cannot do this kind of generalization, because even some kind of organization can say it very fast and very clear and can be even dangerous consequences. There was a publication from 2013, I think, that there was an action trying to reduce malaria in some parts of Amazonas by making a kind of reforestation again. More people die of malaria after this action. So these kind of methods that are very nice to hear sometimes can be even dangerous, okay? This is my guessing about that. Of course, we're still in the debate. There is some kind of consensus. What is the scale at what dilution effect is more or less working? Also, we have other consensus, like, of course, in diseases, in pathogens that are generalists and air transmitted, it's almost impossible to have a dilution effect because the ratio on contact is, I mean, we cannot expect a dilution effect in COVID when we are transmitted through the air. It's almost impossible. So this kind of consensus that start to be, but to do generalization can have this kind of very big consequences at the end. So we have to take with care when we just go ahead, because we cannot deny that in science there is also some kind of ideology, and we have to be aware and, of this kind of messages that we can send to the population, because sometimes are very nice, but can be very naive and even very dangerous, okay? And just to say thank you to all these people that collaborated with me in the last years, my enterprise, my postdoc in Israel, my partner in this kind of articles of divulgation, and the group of Jose Martinez de la Puente. Thank you so much. I don't know if I take a lot of time, more or less. And if you have any question, and I can respond, I will be happy. Thank you.
Thank you very much, Mario. We have time for questions.
Thank you. Thank you, Mario. It was an amazing talk. Like I studied for my thesis, host parasites coevolution. So this was like a second Christmas for me in terms of information. But like, we study more or less the same, but like you use different language, so just maybe these questions are a bit naive, but in disease risk, what is the most heavy factor, virulence or transmissibility? Because there's like a...
this is another of the problems that we are having. I mean, I'm not having. Yeah. I'm not defending the dilution effect like the authors that I think they want to at some kind to win the Nobel Prize. But let's say that there is also a debate on what kind of metrics we have to take in order to really know what is disease risk. Mm-hmm. And also these people like Kevin Lafferty, Dobson, all these kind of people start to state that it has to be related to each pathogen. So at the end we are going back. It's like each system is singular. Yeah. We cannot make some kind of generalization. Of course, there are some kind of common paths that are like in tropical and subtropical regions. This is not working so well because there is a lot of interactions. As I said, not with direct transmission disease. Also, sometimes it doesn't work. Indeed, the same author, Osfelas Kissing, in the first paper, they established 5 points by which the dilution effect can work, and then suddenly everybody forgot about them. They never mentioned it again. But if you go to the first papers, there are 6 or 7 conditions by which dilution effect should work. So I don't know. It's kind of, I can understand it, no? But if you have data to do it, but it's kind of, don't know, not to that we are in this kind of rush to publish a lot, and sometimes we don't take the time enough to let the things to reposar.
Yeah. And like following this, because I'm just curious, like Andersoni, Gervillus Andersoni was the one that never overcome the infection.
No, Gervillus Andersoni is the one that has more infection.
What was the one that never, like it's infected and it's forever?
It's infected and is forever. In our experiment was for 156 days. Yeah. So
I mean, a lot of time. The longest infection.
A lot of time feeding gerbils every fucking day.
Yeah, I'm following this, like the virulence, like the aggression of this of the parasite, since it was also the one. Could you see like he was more healthy than the others? Like the others had a reaction, and then there was like no fitness component. Like they were like, fine.
In the things we measured about the lethal effects or sublethal, we find nothing. But of course, this is lethal or sublethal effects that are in animals that are in the laboratory, are well fed, and all these kind of things. Maybe if you infect Gervillus and Pyramidum in the field with other kind of risk around, they can have some consequences. But is the trade-off of the experiment or under natural condition or in the laboratories? We have to do these things still.
Yeah, it's super interesting. Thank you.
Indeed, what I was saying is that one of the things that we did that we have just published, we're going just to have published, is one of the things we were going to to analyze what is the different relations. But we have some analysis of the pheromones of infected and not infected individual of the 3 species to see if there is kind of attraction in order to facilitate the transmission. That you know that infected individuals that change the chemical communication in order to get susceptible individuals and to get infected about them, the manipulation of parasites. In this case, we have with people from the Museo de Ciencias, José Martín and Pilar López, and we have to do this. There are things that are still have to be answered. Also, we have buccal swab to see if the infection is also in the buccal swabs when they bite one each other, and we have very nice things. We have videos of aggressions between the different species to see how they react when they are infected or not, when they are the owner of the land, let's say. I don't know, I cannot get down the number in English. So all this kind of information we have to analyze it still.
More questions?
Thank you, Mario. It was a really nice talk. I have a question regarding, like, you went a little fast because of time, the stabilized sun result for Mycoplasma. It wasn't clear to me if it follows the same pattern it was following before, or the alteration changed the patterns that you were seeing before.
The patterns are the same. I mean, in the experiment I was testing from here to here, how host community composition can affect Mycoplasma or Bartonella. What we did in the field with this orthophoto is to say, okay, so what is changing the host community composition? And we went over here. So from here to down, the results are the same. Okay. But we have it there that the host community composition can change with these anthropogenic disturbances, more or less.
Okay. Thank you.
Pleasure.
More questions? Anybody else? Come on, don't be shy. Nope? Okay, so thank you, Mario, for your talk, and see you everybody the next week for the next seminar.
Thank you. Look at it.